You've probably been taught, that a cell is basically a tiny biological machine. We all learned it in high school biology,
Well,
right?
absolutely. The classic textbook diagram.
Yeah, exactly. It's got this master blueprint, the DNA, and that blueprint sits safely inside the nucleus, just giving out orders. It tells the cell exactly what shape to take, what jobs to do, who to be. It is a very top-down engineering-style view of life.
It is, and it's comforting, because it's linear. We really like to think of biology as a set of dominoes falling in a perfectly predictable line, starting with the genetic code.
But what if that is entirely backwards? What if a cell's physical shape, or even just how squishy the environment around it is, can boss its DNA around just as much as the DNA dictates its shape?
That is the big question.
Right. So today, we are taking a deep dive into a paradigm-shifting 2024 paper. It was published in the journal Cell by Suzanne M. Rafelsky and Julie A. Theriot. And our mission for this deep dive is to completely reconstruct your understanding of what a cell state actually is.
Which is such a crucial mission because, we are talking about the fundamental unit of life here. You can't break life down any smaller than a living cell. But here's the overarching problem we've run into science has been trying to model cells the way a mechanical engineer models, like a jet airplane.
Which, makes sense on the surface. We have all these discrete parts, so let's just map them.
Right. With a jet, you can use fluid dynamics and heat transfer to build a perfect digital twin in a computer. But living cells aren't jets. They are what physicists call dissipative systems.
Okay. Dissipative systems, meaning?
It means they constantly use energy. They operate far, far away from chemical equilibrium, and they are constantly exchanging matter with their environment. They are messy, dynamic, and incredibly complex in a way that a closed mechanical loop just isn't.
So if the engineering approach is failing us, how did we even get stuck in this trap? Because for a long time, looking at a cell was pretty straightforward. We'd look under a microscope and just classify it by its shape or its job.
Yeah, the morphological era.
Right, like epithelial cells in your skin. We'd call them squamous if they were flat, or cuboidal if they looked like little cubes.
Exactly. We categorized them purely by how they looked and where they were, but then came the molecular era, specifically the post-genomic era. Suddenly, we developed these incredible technologies like single-cell RNA sequencing.
Which is cRNA-seq, right?
Yes, exactly. And we started building massive data repositories like the Human Cell Atlas. And so we shifted from looking at a cell's physical shape to obsessing over its molecular readout.
Which sounds like a massive upgrade. We can suddenly look at the exact genes a cell is expressing at any given second.
It is an incredible technological leap, don't get me wrong, but it led us into a conceptual dead end. The authors of the paper call this the bag of RNA trap.
The bag of RNA?
Yeah. Because humans have about 20,000 genes, scientists started representing an individual cell as a single mathematical point in a massive 20,000 dimensional space. We started assuming that if we just mapped the transcriptomic profile, the exact RNA floating around in that bag, we would completely 100% understand the cell's identity.
Wait, let me make sure I'm getting this. We assumed the parts list is the identity.
Yeah.
But does mapping all those genes actually tell us what the cell is doing?
That's exactly the problem. The paper highlights that mapping genes does not perfectly map function. There is a fascinating disconnect here. Take human stem cells that scientists coax into becoming heart muscle cells in a lab-induced pluripotent stem cell-derived cardiomyocytes.
Okay, a bit of a mouthful, but lab-grown heart cells, got it.
Right. So as these cells mature, their primary cardiac muscle protein physically shifts from one version to another. It shifts from an alpha version, which is coded by a gene called MYH6, to a beta version, coded by the MYH7 gene.
Okay, so if I'm looking at my my bag of RNA, I'd see the MYH7 RNA spiking and think, great, this is a mature, perfectly functioning heart cell.
You would think that, but the authors point out there is literally zero correlation on a cell-by-cell basis between those gene expression levels and how well-organized the cell's actual physical muscle fibers, its myofibrils, are.
Wait, really? Zero correlation?
Zero. The RNA data says one thing, but the physical reality of the cell's myofibrils says another entirely.
That is wild. It's like having an incredibly complex, deep learning AI that can perfectly predict the exact time of sunrise every day for the next 1,000 years.
Oh, that's a great analogy.
Right, sure, the data output is perfectly accurate, but that algorithm doesn't teach you Newton's laws of physics. It doesn't actually explain why the sun is rising. We have all this RNA data, but we're completely missing the underlying rules.
Exactly. We are drowning in data, but starving for fundamental laws. The authors argue we need to Keplerize the cell.
Keplerize, as in Johannes Kepler, the 17th century astronomer.
The very same. Think back to astronomy. Tycho Brahe collected a mountain of incredibly precise datasets on planetary positions, just walls of numbers. But it was Johannes Kepler who looked at that massive dataset and found human-interpretable patterns, Kepler's laws of planetary motion.
Nice.
Right, and only after Kepler found those underlying patterns could Isaac Newton come along and formulate the fundamental laws of physics. Right now, biology has a lot of Brahes collecting massive data sets, but we desperately need more Keplers to find the patterns.
And the paper gives a really cool example of this Keplerizing in action, right, with the lac operon in bacteria, which is just the system bacteria use to digest lactose.
Yes, it's a beautiful example. If you just measure the concentration of the repressor protein involved in that system, the data looks like a chaotic mess. It's scattered everywhere based on binding affinity, how many gene copies there are, just all sorts of noise.
So it looks completely random.
It does, but researchers found that if you plot that exact same gene expression data against a physics concept called repressor fugacity, all that noisy data collapses perfectly into a single, elegant, universal curve.
Wait, fugacity? What is that? That sounds like intense physics jargon.
It is, but think of it like the pressure inside an overcrowded room. Fugacity is essentially a measure of how badly those molecules want to escape and spread out their chemical potential.
Oh, okay.
Yeah, so when you stop looking at just the raw count of molecules, the concentration, and start looking at how those molecules physically want to behave, the chaos suddenly makes perfect mathematical sense.
So if I'm tracking this right, let's do a quick recap. We've moved from historically categorizing cells just by their shape under a microscope to recently obsessing over their RNA as a 20,000 dimensional parts list. And now we're realizing that the parts list alone is a dead end. We have to start looking for these deeper, holistic patterns.
Exactly. Genes alone don't equal cell reality.
Which makes you wonder, what hidden patterns do you think we are missing in human health right now, just because we are so hyper-focused on the genetic parts list?
It absolutely does. Which brings us to the core of the author's proposed framework. If looking at a cell as just a bag of RNA isn't enough, how do we actually measure a cell's true identity? The authors argue we need to look at all cellular observables simultaneously.
Okay, but what does that actually mean in practice? What else are we looking at besides the genes?
They break it down into 4 practical categories. First, you need the molecules, obviously, the molecular census. This is every single molecule in the cell and its exact quantity, not just the transcriptome or the RNA, but the proteome, the lipidome, everything.
Right, the census. But then we also need to know where all that stuff actually is.
Exactly, which brings us to the second category, organization. This is the multi-scale spatiotemporal arrangement of everything in that census.
Multi-scale spatial meaning where it is in space and time.
Right. What is the physical shape of the cell? Where are the organelles located? And we are starting to get massive data sets for this too. Projects like cell painting and OpenCell use fluorescent dyes to map the inner 3D architecture of thousands of cells.
Okay, so we have the molecules, we have the organization. What about what the cell is actually doing?
That's the third, function. These are the dynamic actions the cell takes that are relevant to the organism's survival. Is the cell using directed migration to move toward a wound? Is it generating physical force? Is it actively secreting insulin?
And I imagine the final piece of the puzzle is where this whole thing is sitting, the environment.
Spot on. The cell's environment, extrinsic features. What is the chemical makeup of the fluid outside the cell? How mechanically stiff is the substrate it's anchored to? What neighboring cells is it bumping into?
Wait, molecules, organization, function, environment.
Yes.
M-O-F-E-M-O-F-E. That is a perfect mnemonic to remember it. Think of a cell like a major city. The molecular census is the population count and demographics, how many bakers, how many mechanics. The organization is the city map where the buildings and streets are. The function is the daily traffic and the actual economy running. And the environment is the surrounding geography and weather.
That is a brilliant analogy.
Because if you want to understand why a city works the way it does, you can't just read the population census. You need the whole picture.
You really do. And just like a city, it raises an important question about boundaries. Because in a living cell, these 4 categories form a continuum.
What do you mean by a continuum?
Well, at what point do several individual proteins, which are technically part of the molecular census, bind together to form a large structural scaffolding complex that dictates the cell's physical shape, which is organization? The line blurs constantly. We have to measure the variability across cells, not just the averages.
All right, so let's lock this in. M, molecular census, the parts. O, organization, the structure. F, function, the behavior. E, environment, the surroundings. Next time you picture a cell in your mind, which of these four multi-categories do you realize you've been completely ignoring?
Usually it's everything but the census.
Yeah, but if I picture this city or this cell having these four distinct categories, how do they actually talk to each other? We spent 50 years assuming the census, the DNA and RNA is the mayor of the city pulling all the strings.
And that is the exact assumption this paper dismantles. To understand how they interact, the authors introduce a really elegant conceptual visualization. I want you to look at figure 2 from the paper in your mind's eye. Picture a tetrahedron, a 3D pyramid with a triangular base. It has 4 points, or vertices. Each of those 4 points is one of our Hairy categories.
Okay, I'm picturing the pyramid. Census, organization, function, environment at the points.
Now, connecting those 4 points are springs. The springs represent bidirectional feedback, the push and pull between the categories. A stable cell state is not frozen like a statue. It's constantly fluctuating. It is a jiggling tetrahedron.
A jiggling tetrahedron, I love that.
If the springs connecting the vertices are stiff, the cell state is tightly regulated, like sitting in a narrow potential well. It doesn't change much. If the springs are soft, the cell allows for a lot more variance, a broad potential well. But in both cases, the springs are always pulling the tetrahedron back into a stable, recognizable shape.
Here's where it gets really interesting, though. You said the springs represent bidirectional feedback, meaning they pull in all directions equally. Are you saying the physical shape of the cell or its environment can boss the genetic census around just as much as the DNA bosses the shape around?
Yes.
How does a physical shape change a genetic code?
It sounds like science fiction, but it happens all the time. Let's look at some graded evidence of this feedback. Take human mesenchymal stem cells, HMSCs. These are basically blank slate cells. If you place these stem cells onto a glass plate with large microscopic islands of a protein called fibronectin, the cells spread out flat to grip the surface.
Okay, so their organization changes based on the environment.
Right, and that simple physical shape change induces them to become osteoblasts, bone cells. But if you take the exact same stem cells and put them on tiny islands where they don't have room to spread out, they stay around. And because they stay around, they turn into adipocytes, fat cells.
Wait, hold on. The chemical signals in the fluid around them were identical, right? The only difference was the physical space they had to stretch out.
Identical chemistry. The physical environment alone dictated their entire genetic destiny. And it goes even further. If you put those same stem cells on a soft, squishy substrate, they turn into neurons. Put them on a stiff, rigid substrate, they turn into bone. Intermediate squishiness yields muscle.
Okay, that makes sense on a macrocellular level. But mechanically, how does a physical squish reach into the nucleus and change the genes?
That's where organization directly alters the census. Think about the nucleus of the cell as a porous balloon. If you mechanically put physical pressure on a cell and literally squish it, you physically stretch the nuclear envelope membrane.
Okay.
And when that membrane stretches, it distorts the shape of the nuclear pores, the tiny gates that let things in and out of the nucleus.
Oh, wow. So you are physically opening the gates wider.
Exactly. You stretch them. And this physical distortion changes the rate at which transcription factors, the proteins that turn genes on and off, can passively diffuse into the DNA. You are altering the cell's genetic output simply by applying physical force to the outside of the cell.
That is mind-blowing. But surely a cell can't actually do anything complex or purposeful without its DNA calling the shots at some level, right?
You'd think so, but let me tell you about primary fish basal epidermal cells called keratocytes. If you put them on glass, they just sit there in a perfect stationary circular symmetry. But eventually, through spontaneous, tiny actin-myosin contractions, just physical pulling of their internal protein fibers, they break their own symmetry. They shove all their organelles to the back of the cell, and they just start sprinting forward across the glass.
Just a spontaneous mechanical reaction.
Yes. But here is the staggering part. If you take a microscopic scalpel and cut off a piece of that cell, a fragment that has absolutely no nucleus, zero DNA whatsoever, that enucleated fragment will undergo the exact same state transition. It will break symmetry and purposefully sprint forward across the glass.
Wait, without a nucleus?
Entirely without it. No DNA or gene expression changes are required at all for this massive behavioral shift. The function happens entirely without the census.
So a piece of a cell can essentially think and move purposefully without its master blueprint even being in the room. It really makes you question, where a cell's identity actually lives. It's not just a top-down machine following orders.
Not at all.
It's this dynamic, jiggling tetrahedron where physical pressure The squishiness of its neighborhood and its genetic code are all constantly arguing with each other to find balance.
And that constant argument leads to one final mind-bending example. What happens when the census alters function, but without involving genes at all? Take dermal fibroblasts, skin cells. They have distinct lipotypes, meaning they have different specific sphingolipids in their outer membrane.
The fatty layer on the outside.
Right. Changing those lipids completely alters how the cell receives FGF signaling growth signals, which totally changes the cell's behavior. But if you look at the RNA for the enzymes that make those lipids, there is zero change in gene expression. The genes are totally silent, yet the cell has completely changed its functional identity simply because its lipid parts list shifted.
Okay, so quick recap of this part. The cell is a dynamic tetrahedron connected by springs. Changes in environment or physical shape directly alter genetic expression. And complex behaviors can occur purely through physical mechanics, without any genetic changes. Which leads me to ask you, if a piece of a cell can think, break symmetry, and move purposefully without its nucleus, where does the cell's identity actually live?
That is the philosophical core of this whole paper, really.
It is. Okay, but if these springs in our Mothi tetrahedron are always pulling the cell back into a stable, balanced shape, how does this cell ever permanently change? Like, how does a single fertilized egg develop into a human being? Or how does a healthy cell suddenly warp into a permanent cancer state?
That is the ultimate question. To understand permanent transitions, we have to visualize what the authors call the manifold. Imagine figure 4 from the paper, a massive, ultra-high-dimensional, warped 3D landscape. A really bumpy alien terrain. This is the manifold. And it represents all achievable combinations of our cell observables. Because let's be real, not every combination of proteins and shapes is biologically possible.
Right. Physics still applies.
Exactly. So a stable cell is basically jiggling around in a tiny, comfortable pocket on this bumpy landscape.
Okay, so a state transition is just popping out of one pocket and landing in another.
Exactly. A transition happens when one vertex of our tetrahedron gets pulled so hard, say, by a massive environmental shift that it falls completely out of alignment. It yanks the spring so far that the whole structure snaps into a new stable configuration. The cell pops out of its comfortable pocket and settles into a different pocket on the manifold.
I remember learning a version of this in college called Waddington's landscape. The classic analogy is picturing a cell like a marble rolling down a fixed, paved mountain, choosing different valleys as it goes toward its destiny.
Right. Waddington's landscape has been the standard mental model for decades, but it has a fatal flaw. Cells are dissipative systems, remember?
Right, they consume energy.
Yes. They don't just passively roll down a hill guided by gravity. The authors propose we need to update this to a motivational energy landscape. Because the cell is actively consuming energy and reacting to its environment, the landscape itself changes as the cell moves through it. It has history and memory.
Oh, so Waddington's landscape is like a marble rolling down a paved mountain. But this new holistic model is more like a snowplow, carving its own path through deep snow.
I love that.
As the snowplow moves forward, it literally alters the terrain behind it. It leaves a massive trench, which makes it physically impossible to just slide backward in exactly the same way. The cell shapes the landscape just as much as the landscape shapes the cell.
That is a perfect way to visualize it. And we see these non-reversible transitions in biology all the time. Take a process called EMT, epithelial to mesenchymal transition.
What's that?
This is when a stationary, rigid cell transforms into a mobile, migrating cell. It is critical for embryos developing, but it's also how cancer spreads and metastasizes. Moving from state A to state B is not simply the exact reverse of moving from B back to A.
Because the snowplow already changed the terrain.
Exactly. Or look at the cell cycle when a cell divides to replicate itself. Why does it only move forward? Why doesn't a dividing cell ever just get halfway through and then run backwards to where it started? Because its dissipative motivational energy dynamically alters the probability landscape behind it, effectively erasing the path back. It burns the bridge after crossing it.
That is so cool. So cells move along a restricted manifold of possible states. Transitions occur when the tetrahedron springs are pulled into a new stable configuration. And cells generate motivational energy, altering their own landscape as they transition, creating actual cellular memory.
Perfectly summarized.
It totally changes how you picture the cells in your own body right now. Millions of them are dividing and moving. How does the idea of a cell having physical memory and motivational energy change how you view those dividing cells? They aren't just following a rigid genetic script, they're acting like little snowplows making real-time physical decisions.
And if we step back and synthesize why all of this matters, it really comes down to the future of medicine. If we want to build next-generation therapies, if we want to truly conquer cancer or master stem cell regeneration, we have to stop staring only at the DNA.
We can't just look at the census to fix the city.
Exactly. We must treat the cell as an integrated, holistic, four-dimensional entity that actively shapes its own destiny. We don't need to perfectly measure every single molecule in the universe, but we do need to start measuring the right things across all four MOFI categories, and we will likely need new physics-aware deep learning models to find those hidden rules to Keplerize the data.
It is a total paradigm shift.
There.
And the authors leave us with a mind-expanding thought in their conclusion that I just cannot shake. They point out that this holistic approach doesn't just stop at a single cell. What if we scale it up?
To the tissue level.
Even bigger. If a microscopic cell is a fluctuating tetrahedron defined by its census, its organization, its function, and its environment, couldn't complex tissues, entire human beings, or even whole ecosystems be mapped onto this exact same motivational energy landscape?
Oh, wow.
Are we ultimately just giant fluctuating tetrahedrons carving our own paths through the snow?
It is a profound way to look at life at every single scale.
It really is. Remember how we started talking about that rigid engineering style blueprint, the master DNA giving top-down orders?
Right.
I think it's safe to say that blueprint has been officially shredded today. The reality is far squishier, far more dynamic, and infinitely more fascinating. Thanks for taking this deep dive with us today. Look at the living world a little differently today, and we'll catch you next time.